Good morning, good afternoon, good evening. My name is Bali Palendran, and I'm calling from Stanford University. Welcome to this week's Global IMMUNOTALKS. Today, I have the distinct honor and pleasure of introducing our guest speaker, Professor Menna Clatworthy from Cambridge. Now, many of you know Menna from her outstanding work in understanding the regulation of immune responses, but let me give you a brief introduction. Menna is the director of the Cambridge Institute for Therapeutic Immunology and Infectious Diseases. She is the professor of translational immunology at the University of Cambridge, honorary consultant nephrologist at Adam Brooks Hospital, and associate faculty cellular genetics at the Wellcome Sanger Institute, and a fellow and director of studies of clinical medicine at Pembroke College, Cambridge. Now, Menna is a clinician scientist, so she studied medicine at Cardiff University and then completed a professional training in nephrology at Cambridge and did her PhD in the laboratory of Professor Ken Smith at the University of Cambridge, where she did some very elegant work understanding the mechanisms by which antibody molecules mediate their effector functions through Fc receptors and regulate autoimmunity and infection, the response to autoimmunity to infections. She then completed a Wellcome Trust Intermediate Fellowship at Cambridge and the NIH, gaining experience in advanced imaging techniques in Professor Ron Germain's lab. And Menna is a clinician scientist in the truest sense of the word. So she spends her time between clinical work as a nephrologist and really doing beautifully elegant work in cellular immunology at the very highest levels. So her main areas of scientific interest are understanding the regulation of B-cell activation and antibody effector function, and investigating tissue immunity across organs and disease states. And this work ranges from experimental medicine studies in patients uh, through to basic immunology studies using mouse models. Her scientific achievements have been recognized by election to the Academy of Medical Sciences in 2020, the election to the Learned Society of Wales in 2020, and election to the European Molecular Biology Organization in 2022. So we're in for a treat today. And so, Menna, welcome. Feel free to share your slides. But before you begin, as is customary, I'd like to ask you a question. And the question I'd like to ask you is the following. What unexpected skills or experiences outside of science have helped shape your career?
So thanks, Barley, and also to the other organisers for inviting me. Yeah, so in terms of that question, so as you kind of alluded to there, I'm actually Welsh, so I'm originally from Wales, and part of the traditions there and part of what I did in school is something called Eisteddfods. And these Eisteddfods are gatherings where you meet on St. David's Day, March the 1st, and you have to do things like recite poems out loud to everyone, sometimes in groups or sing songs. And so I really enjoyed and did a lot of poetry recital. And that's something which I think has helped me in terms of public speaking, in presenting science. You'll be able to judge at the end of this whether I'm right in that. But also it's left with me a love of poetry, which is can actually be very instructive. There's what I will say is there's much wisdom in poetry, and I sometimes even in our beginning of year lab meetings share poems with my probably slightly bewildered lab members. So that's my answer to that question.
Wonderful. Thank you for sharing that, Menna. That's truly inspiring. Uh, we're not going to ask you to recite your favorite poem here in Global Immunotalks. And I must say that writing poetry and writing a scientific paper, one has a schizophrenic experience in many ways, right? In some ways. But in any case, welcome. Before you start, I should also mention there will not be a formal Q&A session. If you do have questions, please send them or post them on the Global Immunotalks Twitter account, and we will compile them and get the answers to you. So welcome, Menna.
Thanks, Barry. So you can see my screen, hopefully.
Yes, yes.
Yeah, awesome. Okay. So the title that I've given today is Adaptive Immunity at the Central Nervous System, some borders. And what I briefly wanted to say before I dig in there is as Barley has said, my lab studies tissue immunity across a bunch of different organs, actually. And by studying many organs, there are advantages and synergies to that. But what I want to focus on today, in the interests of a talk that's 45 minutes, is just on one of those, and maybe one that I'm really excited about, which is our work in the central nervous system. So, of all organs, perhaps arguably the brain is the most important. It allows us to engage in activities that make us uniquely human. And I think that's evolutionarily reflected in the effort that has developed in terms of defending the brain. So uniquely amongst organs, it's completely housed more or less by a hard box, so the skull. And as well as that, it's covered by three layers, so the meninges, the outer dura mater, the middle arachnoid mater with a cerebrospinal fluid between it and the pia mater, which lines the brain. And the dura is also interesting because it has these very large venous blood vessels, the dural venous sinuses. And if I show you on the left here, this is an X-ray in a human. You can see the major one. This is the front, the nose, the back. The major one is the superior sagittal sinus, which runs down the middle, and then there are transverse sinuses at the back. And in mouse, we can visualize this with whole mounts, so we can actually take off the whole of the dura from the inside of the skull, and you see similar structures. So the transverse sinuses at the back, and then the superior sagittal sinus. And what's quite remarkable is that much of the vasculature within the dura is actually fenestrated. And the dural venous sinuses are also well connected. They drain blood from the brain, but also from the surrounding structures, so the subcutaneous space of the scalp and also from the skull bone marrow. And the fenestrated nature of this vasculature and the fact it takes its blood ultimately from the circulation, it means that pathogens or any antigens or immune stimuli that are present within the bloodstream could potentially hop off through these fenestrations into the dura. And in fact, we know that does happen. So there are a number of bacteria that can access the dura and cause meningitis. And I've listed the top 3 causes in immunocompetent individuals there. And when we think about the importance of this as an evolutionary pressure for driving the way this system is set up, the immune system around the dura is set up, even in the current antibiotic era, neurological infections in children amount for around 5% of deaths in the under 9-year-old age group. So this is a much more potent evolutionary pressure because it determines whether you will or won't pass on your genes than say, autoimmunity or neurodegenerative diseases. So I would argue that understanding how the CNS defends itself against infection is really instructive in thinking about the rules of how this tissue immune system is set up. And so one of the, I suppose, revelations in the last decade, when I was in medical school, the notion was that the central nervous system contained no immune cells, bar microglia. But we now know that actually the dura in particular houses a whole variety of immune cells. And this is an atlas that we put together of published healthy border tissue data. So you can see there are both innate and adaptive immune cells. And there have been some pretty interesting studies. So, for example, from Yoni Kipnis on the functionality of some of the T cell subsets. But what my lab are interested in, as Barley's described to you, is humoral immunity. And the purpose of humoral immunity, to remind you, of course, is to generate antibodies. And the central cell components are B cells, which express a surface antibody or B cell receptor that on engaging antigen can become activated and differentiate ultimately to form a plasma cell, which secretes these antibodies. And we know that there are different antibody isotypes in different regions of the body. Those isotypes are determined by the nature of the heavy chain. So if you have a gamma to heavy chain, you have an IgG antibody, and we know, if I took your blood, the dominant antibody there is IgG. It's very pro-inflammatory. Largely speaking, it can fix complement and also activate many innate immune cells by binding to Fc gamma receptors. Now, in contrast, at mucosal surfaces like the gut, the dominant antibody isotype is IgA. And this is much less inflammatory. It doesn't fix complement. And actually, in the gut, acts to sort of enchain bacteria, keep them away from the epithelium because everything gets stuck within the mucus layer. So they have a very different purpose. And so we came to this a number of years ago now. In the first part, I'll skip through quickly because it's been published a while. This was taken on by Zach Fitzpatrick, who's actually an American PhD student who came to me via a PhD scheme that works between Oxford and Cambridge and NIH. And so Zach very simply initially did some microscopy, so some of that whole mount imaging that I showed you, to look at where B cells and plasma cells were found within the dura. And what we found, so using BLIMP1, so a transcription factor that's characteristically expressed by plasma cells, CD138, a surface marker of plasma cells, we could find that plasma cells were largely speaking found along the walls of the dural venous sinuses and located adjacent to vasculature. And that was also true of other immune cells and B cells too. So I sent Zach away to look at what antibody isotype those plasma cells may be producing, thinking that it was likely IgG, given this was an internal organ. And I was surprised to find that predominantly these cells were IgA positive or IgA secreting. So you can see here, this is the front of the mouse, so just above the nose, the superior sagittal sinus, the transverse sinuses. In red, we have the IgA plasma cells that you can see are also 138 positive. And we found that this feature was actually conserved across species. So I was able to get some human dural samples from neurosurgical colleagues, and we were able to find IgA 138 positive cells in human dura. And this raised the question to us, sort of thinking that IgA is normally something that's found at mucosal surfaces, and particularly the gut, whether there might be a relationship between plasma cells and B cells in the gut and in the dura. And we looked at this in a number of ways. So, for example, we did some studies in germ-free mice showing that these cells were almost absent within the dura. We looked at some antibiotic treatment, and again, this reduced the number of dural IgA secreting plasma cells. But we also did this experiment where we took a centimetre of small intestine and the whole of the dura and did some B cell receptor sequencing. And we've plotted these as these BCR network plots where each dot is a BCR sequence and it's joined to a related BCR by a line, and depending on the number of mutations, that will determine the length of that line. So you can see some B cell clones here. So in the top, the meninges, if we focus on the IgA only, there are some large clones here. And the ones that are coloured, we were also able to find family members within the small intestine. And even though we only took a small part of the small intestine, so a mouse small intestine is about 30 centimetres, we took just 1 centimetre, we were still able to find 20% overlap in the BCR clones. And we also found that although in our mouse house at that particular point in time, the dominant isotype within the dura was IgA, if you caused intestinal inflammation using DSS colitis, you could switch that to IgG, and also the gut microbiome could alter the antibody isotype. And if we looked at some of the BCRs, largely we found that many, so this is the germline BCR, and then this is the number of mutations, and this is BCRs we could detect in the small intestine or in the pink in the meninges. Many of the small intestinal BCRs were proximal in the family tree to these distal ones, suggesting that they originate here and then end up in the dura. And we can test this more directly using a photoconversion model. So we use the kaidi mice, and kaidi is the Japanese word for maple. So these mice ubiquitously express green fluorescent protein, which you can convert with a UV light to a red fluorescent protein. You can detect this by flow cytometry. So what we did with our collaborator, Dave Withers, he actually had an endoscope, so a colonoscope that we could put into the mouse large intestine and shine the UV light. And then we could photoconvert the cells within the wall of the intestine and then track them. And even in homeostasis, 24, 48 hours after, you can identify in the extravascular space of the dura KRED positive B cells. And we could also actually find some of these in the brain too. So B cells are tonically trafficking from the gut to the dura and the brain. And so what's the functional purpose of this? It sort of seems like a lot of hassle to go to. So we proposed that this was because you needed that IgA at this vasculature as a defense mechanism. And so to test this, we began to use some intravenous challenges. So in this case, we're using intravenous candida, so in purple here. And you can see this is an area along the wall of superior sagittal sinus, and now IgA is in white, that actually the IgA is, that you can see some plasma cells here, but secreted IgA is actually bound to and entrapping, just as been seen in the intestine, these candida. And so what we needed to do to test functional importance was to actually deplete those plasma cells. And this was a tricky challenge, because ideally what we needed to do is to deplete plasma cells specifically in the dura and leave plasma cells elsewhere untouched. And the way that we came up with doing this was actually based on the knowledge that proteasome inhibitors are used in multiple myeloma, so a plasma cell malignancy. And we used one of these, bortezomib, and actually put it within a hydrogel and use the anatomy that I showed you, the fact that if you put something subcutaneously in the scalp, actually it will drain down through the dura and into the dural venous sinuses. And remarkably, this worked really well to deplete the plasma cells just within the dura, leaving those in the spleen and bone marrow untouched. And then if we repeated that experiment, challenged with intravenous candida in the absence of those plasma cells, we could no longer see that the candida was held up at the borders, but rather when we looked for the GFP candida within the brain, we could find it there. And unsurprisingly, if the candida gets into the brain of the mouse, they don't do so well. So this is pretty remarkable, defining that humoral immunity initiated and educated or primed within the gut ultimately led to defense at the dural venous sinuses. And when I was thinking about this paper and writing it up, in fact, it makes perfect evolutionary sense. We house, I think the estimate is something like 13 trillion microbes within our gut, and we need to do that because we need them for a bunch of different things, but that comes with a price. They can and do sometimes enter our bloodstream. And this is Public Health England data showing that the commonest cause of bacteremia is actually E. coli, happens at about 65 per 100,000. If you ask the number of times, and the data from Public Health England is just for gram-negative meningitis, not just E. coli, the number of times that ends up with a meningitis is actually tiny. So this defense system, I would say, totally makes sense and it works. So if we think about the organisms that do overcome that barrier, so actually many of them are respiratory pathogens or colonizers, so Neisseria, strep and Haemophilus. Although they're a mixture of gram-positive and gram-negatives there, one of the features that's known that they have in common is actually all those organisms produce an IgA protease, so they can overcome this barrier. And we sort of had a little chat in this opinion piece in JXMed about really why it makes sense to link CNS and gut immunity. So one of the things that we noticed when we were looking at our B cell receptor sequencing data from this project, and is that in some of these family trees, we could find further elaboration, so further somatic hypermutation that was specific to the meninges, and indeed some B cell receptor families where the elaboration was entirely in the meninges. And this raised the possibility or the question of whether, in fact, there were structures within the dura that could potentially support a germinal center response where somatic hypermutation could take place. And in fact, when we went back to some of the images that I showed you, what we had noticed pretty early on actually is reproducibly, particularly here. So this is at the front of the dura, so immediately above the olfactory bulbs and therefore above the nose, the cribriform plate and the nose of the mouse. We saw reproducibly these lymphoid aggregates. And often we would see them along the transverse sinuses, and as mice age, actually more of them along the superior sagittal sinus. And so we began to characterize these in more detail, and particularly the one at the front of the dura here, which we called the olfactory or rostro-rhinal hub based on the veins that were around it. And these structures contain not only B cells, but actually also T cells here in blue, and antigen or potentially antigen presenting cells, or certainly myeloid cells that were CD11C positive. And we could see that aggregates, lymphoid aggregates within these structures increased with age, as shown here. And they also contained or were surrounded by lymphatic components and stromal cells, so some live one positive vessels near some of these aggregates, and some podoplanin positive cells consistent with stromal cells that we might see within secondary lymphoid structures elsewhere. And when we characterize the B cells in more detail, we found that at the center of some of these structures were GL7 positive, proliferating, so Ki67 positive cells that we would take as germinal center B cells elsewhere. And by flow cytometry, we could identify GL7 fast positive, double positive cells that again increase with age. And some of these structures on the outskirts of these clusters, we could then find CD138 positive, IgA positive plasma cells, and actually even IgG positive plasma cells too. And we did some staining for some of the potential survival factors for B cells and plasma cells, and could find some local BAF production there too, potentially supporting these structures. So we named these DOLT, so dural associated lymphoid tissue. And again, we went this time to some post-mortem samples that one of my neuropathology colleagues, Kieran Allison, was able to access from the brain bank here in Cambridge. And we could find, again, often adjacent to vasculature within the dura. So we were particularly looking above the cribriform plate and in the frontal dura. So aggregates of immune cells that included B cells, plasma cells, T cells, and myeloid cells. So another example of one here. So we think that these structures are a conserved feature across species. And so my collaborator here, so Doreen McGavin at NIH, was able to do these really amazing sort of mouse CT and to render these images so you could view the vasculature within the head and within the dura, and we could identify the area where our DOLT, so particularly this olfactory or rostro-rhinal hub, sat. And when we were looking at this vasculature, so I mentioned to you that there are descriptions that the vasculature is fenestrated, and so we wanted to know specifically the vasculature around these lymphoid aggregates. So we did some staining. So CD31 is obviously a vessel marker. PV1 is a marker of fenestrated vessels, and then CD45 here. And you can see nicely that there's basically a network of vasculature that's fenestrated. And within this, there are pockets of immune cells. And we're going from left to right here in the olfactory hub. And therefore, unsurprisingly, perhaps then, if you inject a circulating bead, for example, the place where it comes out is exactly within these hubs. And we could see, so whether you're talking about 2 micron beads or 0.2 micron beads, where many more come out, that they are found outside of blood vessels within these hub regions. And we used dextran and then some staining to show actually it's myeloid cells. So, for example, macrophages within these structures are able to take up these circulating antigens. And actually, the B cells within these structures are able to respond to circulating stimuli. So here, if we give intravenous LPS, You can see that the B cells increase in number, some of them proliferate, and actually they upregulate activation markers like CD86. So I propose then that these areas, because of they're surrounded by fenestrated vasculature, are capable of sensing. They're a barometer. They can sense the local circulating inflammatory state. And because of the position here, so I emphasised to you that, you know, the largest hub that we could find was above the olfactory bulbs, and we could often actually find immune cells going down the falx here and to some smaller aggregates that were literally sitting right above the cribriform plate. And so we wondered actually if these structures would allow immune cell responses in the dura to nasal challenges, so to local nasal challenges. And so to test this, we challenged mice with vesicular stomatitis virus intranasally. And you can see following that there was an expansion in the number of B cells. So you can see these clusters forming. And you can see we were also able to find an increase in the number of CXCL13 expressing stromal cells there. They're obviously orchestrating these structures. So we could also find that there were CD4 T cells within these structures, including IL-21 expressing CD4 T cells. So, you know, really suggesting a germinal center response where there were T follicular helper cells providing help to that response. And we also transferred intravenously some antigen specific, so some VSV specific B cells, so in green here, and found that these homed from the circulation to these DOLT structures. And following VSV challenge intranasally, you could see them, those antigen specific cells there proliferating. We also did some single-cell RNA sequencing, so specifically on these DOLT structures. So we kind of harvested the whole of the dura and then cut off the hub region at the front, generated single-cell suspensions. And we did that both in homeostasis and following VSV infection. So this was all of the cells that we got, and then specifically looking at the B cells, one interesting thing that we found clustering in with the TFH were these BT multiplets that contained germinal center B cells and T cells, and we think these are actually, you know, biological multiplets from interacting cells that are there at the time of harvest. So if we compare the proportions in these structures in naive versus VSV, you see a substantial expansion of the TFH post-challenge, as well as plasma cells and these TB doublets, these GCTB doublets. And if we look at between the naive in grey and then VSV here in this blue colour, if we do a cell-cell interaction analysis based on ligand receptor expression in the two cell types, you can see post-VSV there's an increase particularly in IL-21 and also in other co-stimulatory molecules in terms of those interactions. So we were also trying to image the plasma cells that could be generated from this, and we see sort of an early expansion there by day 7 of both IgA and IgG secreting cells that then make their way back along, potentially spreading along the dural venous sinuses that we quantified there below. And we did similarly some B cell receptor sequencing within the DOLT, so you can see in the naive situation the largest clones that we detect when we look at our IgA clones. There are also some IgM here in brown. But at 1 week following VSV challenge, you see an expansion, particularly of these IgM clones. So that makes sense. This mouse hasn't seen VSV before, so you initially get an expansion of IgM. And then by week 3, as we'd seen in that imaging data I just showed you, what we see then is class switching to a number of different IgG subtypes, which you can see there. And if we quantify that switching is present there in the DOLT, and this is not something we see in skull bone marrow. So if we look at some of these individual BCRs, what you can see that within these structures within DOLT, there's quite substantial somatic hypermutation, so an elaboration of these clones. And in response to reviewers' comments on this, one of the things that we did was to define some of the cues that are required to get B cells into these structures. And so if we treated with an anti-LFA1 VLA4 antibody prior to VSV challenge, you can almost negate the development of these structures and of the presence of GCB cells. So, this is like other lymphoid tissues in that circulating cells are able to come in intermittently, and if there's an appropriate antigen and they get activated, they'll be retained there to form germinal centers. But in fact, showing that these structures can completely independently support this response, what we did was an experiment where we started a GC by challenging intranasally with VSV, waiting for the germinal centers to form, and then from day 6 treating continuously then with the LFA1 VLA4. So there's no new input here. Whatever is happening has been started and these structures are supporting it. And we saw no difference between isotype and the anti-LFA1 VLA4 in the class switching that we saw, or in the somatic hypermutation. So suggesting, these structures can really support, things that we would associate with GCs. So to test the functional importance of this, what we did was to then use an anti-CD40 ligand antibody, and we gave this subcutaneously via the scalp, and you could see that this nicely reduced the number of GCB cells that were present within these structures following VSV challenge. And yeah, you can see some of that here. We also found a reduction in the number of IgA and IgG positive cells within the hub. And then a subsequent increase in the amount of VSV that we were able to detect within the brain. So these are potentially functionally important to develop local adaptive immunity against nasal challenges. So we published this last year, and I think it's sort of very important conceptually. These structures, I should say that TLSs have been described in the leptomeninges, so that is in the pia and arachnoid layers, in the context of chronic inflammatory diseases like MS. But actually what we're describing here is within the dura in homeostasis. There are already these rudimentary lymphoid structures that are ready to respond both to circulating challenges, but also to a local nasal challenge. I suppose this kind of in some way challenged the idea, or part of it challenged the idea of what those hubs are there for, because certainly these dural lymphoid cells are helpful in defense, as I've showed you, but actually they also provide a way for the central nervous system to survey what is going on in the circulation. And so I would sort of propose conceptually, when we think about how the brain gets information, we largely think of neurons where there's a direct physical connection between something at the periphery and the brain itself, and those signals are carried via neurotransmitters. But what I'm saying here is that these aggregates of lymphoid cells and lymphoid cells along these fenestrated vasculature actually provide a way for potentially the brain, as I'll come to, of sensing what's going on in the circulation. But not just that, these structures actually receive immune cells that have come from the gut, so that conveying immunological information from there. And I showed you published data on B cells and plasma cells and the evidence we have that they move from the gut to the dura. But what we've been looking at more recently and is unpublished is whether the same rules hold true for CD4 T cells. And so what we did initially was to set up DSS colitis in these mice. So the dextransodium sulfate is an osmotic colytogen that breaks down the epithelial barrier, causes widespread inflammation and the generation of actually Th17 and Th1 cells within the gut. And so what we did was produced the SS colitis, and then I think this was about day 14, we took the dura. We gave an intravenous CD45 antibody before harvesting the mice, so we can identify cells that were within the vessels or outside and within the extravascular compartment. We were then able to identify alpha beta T cells that express raw gamma T, so i.e. the Th17 cells. And those were rare in control dura, but expanded with DSS colitis. And if we look at intravascular versus extravascular and compare colon, dura, and spleen, we can see that the dura actually mirrors the increase that you find within the colon. So DSS colitis is a bit of a bomb. It's, as I said, totally disrupts the epithelium. So we looked at something more specific. So Citrobacter rodentium is a bacteria that also induces Th17 cells within the intestine. And again, we looked following Citrobacter rodentium, I think this is day 21, alpha beta T cells within the dura, we can find an expansion of Th17 cells. And we actually here looked at cytokine secretion, showing that these are principally IL-17 producing. And again, the colon and dura look pretty similar. So the dura is mirroring the colon. So we've gone on to look by microscopies to where these T cells from the gut are localizing. And this is actually an image of the hub area, so the olfactory hub area, and we find that raw gamma T positive CD4 T cells are found exactly within that region. So what I showed you for B cells and plasma cells is true for CD4 T cells. So that information that's gleaned about what's going on in the gut is also trafficked by T cells to the dura. And data that I'm not showing you, we've shown that the same thing is true if you give a Th2 inducing infection. So we've used schistosomiasis, you see Th2 cells in the dura. Th1, we use salmonella, you see the same thing. We've identified the cues that are supporting that in terms of chemokines and cytokines, and that this forms a lasting memory that can be recalled in the mouse even 6 months later. So to sum up, I think this is exciting because particularly the CD4 T cells I've showed you will produce or can potentially produce cytokines, and these can alter brain function. So there's a whole bunch of work, not from me, but from a number of other groups that have shown that cytokines can influence mouse behaviors, so short-term memory, anxiety, social behaviors. And actually this, putting these two things together helps resolve maybe a number of clinical observations around why, for example, patients with inflammatory bowel disease have an increased risk of anxiety and depression. So with that, I'll stop up and just thank those that have been involved in this. So in the IgA story, I mentioned Zach and also my co-senior author, Dorian McGavin. Zach actually honestly did the lion's share of the work involved in this paper. The second paper, again, Zach led on this. So two bioinformaticians in my lab led the work around the B-cell receptor sequencing and antigen receptor sequencing, and Dorian and a number of people in his lab too. So if you like beautiful images of a bunch of different tissues, then check out our Instagram page, and I'll look forward to receiving your questions via Twitter. Thank you very much.
Thank you so much, Menna. Such a beautiful talk. Really appreciate it. As I mentioned, there will not be a formal Q&A, so if you do have questions, please use the Twitter link in the chat box to ask your questions. We will compile them and forward them to Menna and then provide the answers to you. Let me remind you that next week, Global IMMUNOTALKS speaker will be Ignacio Melero, and so please join us then. But in the meantime, have a great week. This concludes this session. Thank you. Bye.
Thanks all. Have a good day.